Bismillah Rahman Rahim. As requested by one of the students on the YouTube comment section, I am posting this video of November 21, paper 5-1. As you all know, this paper is for 1 hour 15 minutes, and you need to be very careful. You need to read the whole question first and then start planning how you want to answer the question. This is a very important point because sometimes you start doing the part A and then you realize that in the part B they're asking something which you're writing here in part A. So please be careful about this. Baker's yeast Saccharomyces cerevisiae has two metabolic pathways for the production of ATP: aerobic respiration and fermentation. Oxygen is not used in fermentation. Fermentation is another name which we use for anaerobic respiration. These pathways are summarized. So they've given you the pathways: aerobic respiration, glucose to pyruvate to acetyl CoA to carbon dioxide and water; fermentation, glucose to pyruvate to ethanol. Some students were researching details of the respiration of yeast. In their research, the students found information about the Crabtree effect. This effect occurs when yeast is kept in high concentrations of glucose. The yeast gains sufficient ATP from substrate-linked phosphorylation during glycolysis. Yeast does not carry out Krebs cycle or oxidative phosphorylation and therefore does not use oxygen. The student wanted to investigate the effect of temperature on the activity of yeast and decided to use yeast solutions in which yeast obtain all of its ATP by fermentation. The students made a suspension. Remember the word suspension. Why? Because yeast is a cell. It cannot dissolve in it. It has a phospholipid bilayer. There's a cell membrane. There's a chitin cell wall. So the students made suspension of yeast from 1 gram of dried yeast and 25 cm cube of water. The suspension was left for 2 hours at 20 degrees Celsius. I always say you must know the difference between solution, suspension, and emulsion. The suspension was left for 2 hours at 20 degrees Celsius. After 2 hours, 25 cm of the glucose solution was added to the yeast suspension and the mixture was stirred. Some of the mixture was added immediately to the apparatus shown in figure 1.2. The students measured the volume of carbon dioxide produced by the yeast fermentation. So now 1 gram of yeast, 25 cm of water, left for 2 hours at 20 degrees Celsius. These are things that they have controlled. And after 2 hours then 25 centimeter of glucose was added to the yeast and the mixture was stirred. Mixture was stirred. Some of the mixture was added immediately to the apparatus and the student measured the volume of carbon dioxide. So please understand this. Then we look at the apparatus. There's a stopwatch, there's a thermometer, there's water, there's a water bath, sort of a water bath. This is the water bath. Then there's a gas syringe, and this is the yeast suspension and the glucose solution. State the independent variable and the dependent variable. So these are things that you can never lose marks on. Everybody must be very clear. Independent is what you have decided to change, and that is the effect of, you're going to study the effect of temperature. So the different temperatures that you take, independent variable will be the temperature. What is the dependent variable is what are you going to measure, not the process. You're not measuring, yes, you are in a way measuring the rate of fermentation, but what are you measuring? You're measuring the volume of carbon dioxide. So do not name the process, you just write what you are measuring. I am measuring your academic progress, but what am I doing? I'm giving you a monthly test, and after 6 months I am going to put all that monthly test together and then show it to you that this is your progress. So what I'm measuring your academic progress, but how am I doing it? I'm doing it by giving you a monthly test. So how is the dependent variable being measured? Volume of carbon dioxide collected. So this gets you to 2 marks. If you said temperature and volume of carbon dioxide collected, or you could even say the time taken to collect a fixed volume of carbon dioxide, like 10 cm cube of carbon dioxide. Please remember, gas and liquids are measured in cm cube, solids in grams. Explain why the student stirred the mixture of yeast and glucose before adding it to the tube in the apparatus. Why would we do that? Why do we? I ask you to make a cup of coffee. Why do you stir my coffee? So that the sugar in it, if I've added sugar and if I've added the coffee granules, so that they mix uniformly, which is called homogeneously. or you can say completely mixed, or you can say evenly mixed. So you ensure that the glucose and the yeast are uniformly distributed. Now, some of you will struggle with English, but I'm sorry, you can come up with something very easy in English as well. Evenly mixed, completely mixed. So any of these wordings would have been correct: completely mixed, evenly mixed, uniformly distributed, and this would get you your 1 mark. Then coming to part 3 of the question, describe a method using the apparatus setup as shown in figure 1.2 that the students would use to find the temperature at which yeast carries out fermentation at its maximum rate. So in a way, it's finding out the optimum temperature. Your method should be set out in a logical order, be detailed enough for another person to follow. You should not include details of how to make the yeast suspension or how to set up the apparatus. And usually you will do the same, write exactly the same thing, not reading the question properly. So use a minimum of 5 different temperatures. Then you state the range of temperatures, like you say 25, 30, 35, 40, 45. Then you use a fixed volume of yeast glucose mixture. Then you use a thermostatically controlled water bath to maintain the temperature, and then you leave the mixture to equilibrate to each temperature. So first you leave it to equilibrate to each temperature, and then you start measuring the volume of carbon dioxide. So you measure the volume of carbon dioxide, say in 1 minute. Use a control. So what do you do is you either replace the yeast with water, or you can use boiled yeast, or then at least 3 minimum repeats and calculate the mean. Do not write average. It's a different meaning. Average means something else. Means means something else. So calculate at least to 5 repeats and calculate the means. Then you repeat the investigation with narrow range of temperatures. Now, what does this mean? Now, I want you to understand this. You see, usually when we get a temperature graph, we get a graph like this, right? And this is the rate of enzyme action. Now, how do we get this graph? Now, for instance, look at it. If I only had these temperatures, if I had only used this temperature and a temperature here, and then a temperature here, and then a temperature here. So if I had plotted this graph, then I would get a graph like this. But if I have not used I've used a temperature here, a temperature here, and then a temperature here, and then. So how do you talk about this? You how do you write this? You'd say use repeat the investigation with a narrower range of temperatures. Like say if this was 20 and this was 30 and then this was 40. So you need it in a narrower range of temperatures, like you should have taken 20, 25, 30, 35, 40, 45. But you see, you now that you know it is something between 30 and 40. So then you can take a narrower range between 30 and 40, like you can have 30, 32, 34, 36, 38, and 40. So if you know that, then you can of course help it to become easier for you to really find out the exact optimum, because if you then have proper points on it, and then you have, so then you would know exactly how the graph is going to go. It would go like this, and then like this, and then like this. So please understand this. What do I mean by repeat the investigation with a narrower range of temperatures around that which seems the one giving the maximum rate? Then, of course, as I tell you, you always talk about the risk, hazard, risk, and precaution. So it's a yeast, could be allergy, use gloves or mask or goggles. And then, of course, you can also say, because it's an enzyme, practical, maintain a constant pH using a buffer. So as you can see, any 7 format, use a minimum of 5 different temperatures. Then you use a suitable range like 25, 30, 35, 40, 45, and please write degrees Celsius. You must write the units. Then known or a standard volume of yeast glucose mixture, 5 ml, 10 ml. Then how do you maintain a constant temperature? You use a thermostatically controlled water bath. So you gave me something about that. You got a mark for that. Then leave mixture to equilibrate to each temperature. So you wait for it, and then of course you leave it to equilibrate for each temperature. Then you leave it for say 2 minutes, a stated time, and then you measure the volume of carbon dioxide. Then use a control either using boiled yeast or water replacing the yeast. Minimum, I don't like this. I say always repeat 5 times for each temperature and calculate mean. That's the important word that you're supposed to be talking about. Repeat investigation with narrower range of temperatures around which seems the one giving the maximum rate. Then low risk or medium risk, I always say, because the yeast and allergy gloves and all that. Then you can use a buffer to maintain a constant pH. That is something that you can always write for enzyme practicals. And part 4 of the question is complete the sketch graph to predict the results that you would expect from the method that you've given in part 3. Include axis labels with units in your answers. So volume of carbon dioxide can either be in centimeters. Now coming to the B part of the question, now this is usually very, very challenging, so you read it very carefully. Scientists have discovered that some other species of yeast show the Crabtree effect. The Crabtree effect means that in high concentrations of glucose and with oxygen present, pyruvate is not metabolized in mitochondria, so aerobic respiration does not occur. Yeast species that are able to maintain their levels of ATP production when conditions suddenly change and no oxygen is available show the Crabtree effect. An investigation compared S. cerevisiae and yeast Candida tropicalis to see if this other species also shows the Crabtree effect. 3 flasks containing suspensions of were initially kept in different conditions for growth. After a set time, the yeast in each flask was transferred to a flask containing a fresh medium with high glucose concentration and no oxygen. All other conditions were standardized. The same procedure was repeated for C. tropicalis. The conditions for growth and the experimental conditions in the flask are summarized on table 1.1. So flask 1, high glucose, no oxygen. Experimental conditions, high glucose, no oxygen. Conditions for growth, high glucose with oxygen, high glucose concentration with no oxygen. So conditions for growth in flask 2, but in experimental conditions it was like this. So flask 3, water with oxygen, and experimental conditions, high glucose concentration with no oxygen. Now, samples taken from the flask kept in the experimental conditions were put into the apparatus shown in figure 1.2 to measure the volume of carbon dioxide produced by the two yeast species. The results are shown in figure 1.3. The final volume of carbon dioxide produced by the sample of yeast from flask 1 at 30 minutes is shown as the maximum volume of 100%. In figure 1.3, all other volumes were calculated as a proportion of the maximum. This allows a direct comparison to be made between the two species. So, final volume flask 1 at 30 minutes is shown as a maximum, whatever it was, that is considered 100%. Say it was 20 cm cube or 30 or 40, that was considered as 100%. All other volumes were calculated as a proportion of the maximum. So like for instance, if it were 20 cm cube in flask 1, that is 100. So in the next, if it's 10, then it would be 50%. So all the other molecule proportion of the maximum. This allows a direct comparison to be made between the two species. So we're talking of flask 1 at 30 minutes was the 100% was shown as a maximum volume of 100%. So please understand this. Now we have a very complex graph. So time after transfer to experimental conditions, this is one. Then time after transfer to experimental conditions in minutes. So percentage of maximum volume of carbon dioxide collected, percentage of maximum volume of carbon dioxide collected. This is the Cerevisiae and this is the Tropicalis. So these are the two different ones. I was very looking at the graphs, very interesting to look at the graphs and see how it is different. Flask 1, compare flask 1 with flask 1, is nearly the same. Flask 2 here is this one. This is a little very, you see it's here, this yellow graph. Let me show it to you. And then this one is, wow, that's really different. And then we have the flask 3 here, and then you have the flask 3 here. So it really shows you a lot of very different. Let me show you this as well. And this, of course, making it colored makes it all very easy for you to look at the comparative analysis of it. Right? So we've got very interesting rainbow colors, green, yellow, red. That's not rainbow actually. So the two different chungered yeast. And let's look at the question. State and explain. Wow. State and explain the evidence in figure 1.3 that supports the idea that Tropicalis does not show the Crabtree effect. It says does not show the Crabtree effect. Now you can see that flask 2 provides us the evidence. Why does it provide us the evidence? Is C. tropicalis there is lower CO2. So in C. tropicalis there is lower CO2. This is 65% and this is 93%. So you gave me figures for that because this is a percentage of the maximum. So flask 2 is lower CO2 production, 1 mark, and then flask 2 is 65% versus 95% would get you another mark. And the third mark would be when you transfer it from conditions with oxygen, with no oxygen, then they have to use fermentation. So the fact that when you transferred it, when you transfer the yeast from conditions with oxygen, then as compare it with the one with no oxygen. So anaerobic, but C. tropicalis does so at a much slower rate, so less carbon dioxide is produced. So tropicalis is producing less carbon dioxide, so that means the rate of anaerobic respiration is much less because there is no Crabtree effect. You see, you have to understand what was the Crabtree effect. So as there is no Crabtree effect, so there's less carbon dioxide.